Today we're learning about independent assortment so we can predict the inheritance of multiple traits simultaneously, and our success criteria is being able to make these predictions using unlinked traits. So Mendel didn't just observe a single trait at a time. Mendel actually observed the relationship between the inheritance of multiple traits. So one thing that he did the observation for is he made the observation of the relationship between seed color in the pea plants and seed shape. In the pea plants, yellow is the dominant color and green is the recessive color, contrary to what you might think, because often we buy green peas. Note here that I've used the letter G instead of the traditional letter Y, simply because G is easier to distinguish between the lowercase and the uppercase. Y it can be difficult sometimes, so I don't like to use it. But typically, because yellow is the dominant color, we do use letter Y for seed color, but it doesn't really matter. For shape, big R, round, is dominant, and little r, wrinkled, is recessive. There are 4 ways you could combine these two traits together. You could get yellow round, yellow or green and round, yellow wrinkled and green and wrinkled. But what we really want to see, since yellow and round are together in the parental generation, and so are green and wrinkled, we want to see if those will split up or if they'll stay together in the F2 generation. Now, we don't care as much about the F1 generation because we know that we have purebred dominant and purebred recessives. We know we're going to get heterozygotes that are yellow and round in F1. So the real telling thing about inheritance is that F2 generation. What happens? Do we only get yellow and round peas and wrinkled and green peas? Or do we get all 4 possible phenotypes? Well, what we do see is all 4 possible phenotypes. The shape and the color genes do not influence one another's inheritance. So when we want to predict 2 traits at a time, which we call a dihybrid cross, we can't just do a 4-square Punnett square anymore. We actually need a 16-square Punnett square in order to correctly predict this inheritance. What we also see is that the ratios change. The phenotypic ratios used to be a 3-to-1 ratio, but now what we see is a 9-to-3 to 3-to-1 ratio. Important thing to remember though, 9-to-3 to 3-to-1 only applies when you're breeding 2 heterozygous hybrids together. It is possible to do a Punnett square not with 2 identical hybrids. The 9-to-3 to 3-to-1 ratio only occurs when you are breeding genotypically identical hybrids together in a dihybrid cross. The 9 represents the 9 of 16 offspring who display the dominant phenotype for both traits. The 3 represents the 3 of 16 offspring who show the dominant for one phenotype and the recessive for the other. The other 3 is then the inverse, showing the other dominant phenotype and the other recessive phenotype combined together. And then that 1 out of 16 is the group of offspring that only show the recessive for both. So what that led Mendel to conclude is that the genes are not influencing each other's. The traits aren't linked together, they don't matter, and we call this the law of independent assortment. The traits not on the same chromosome, important proviso, not on the same chromosome, traits not on the same chromosome will inherit independently of each other. Now, this is one of the cases where, again, Mendel got a little bit lucky because he happened to pick traits that happened not to be on the same chromosome, and he had no way of knowing that. But he did, and we figured out that traits will inherit independently of each other. And we'll talk more about chromosomal linkage later on. This is actually an advantage for us, a big advantage for us in sexual reproduction, because it means that different alleles are constantly being recombined with each other. So we can see more variation, more combinations and possibilities, which improves a species and a population's chances of survival in the case of a crisis.